So hi guys, welcome back to my new video. So today I'm going to talk about B cell activation. So this is a gist of B cell activation which I'm going to talk about. So this is a flowchart, okay, for that. So we start with HSC. Now what is HSC? HSC stands for hematopoietic stem cell. So you can remember it as this is the mother cell of all the kinds of cells which are formed. And if you see the ratio, okay, hematopoietic stem cells, they are present in the bone marrow. Out of 50,000 bone marrow cells, okay, you will get one HSC. So this much is the amount of this is the ratio, okay, of HSC is to bone marrow cells which you'll get. So just imagine how much amount you have in your bone marrow, and they are going to proliferate and they are going to differentiate in case when you are having serious kind of injury. Okay, let's say you met an accident or your body is in a critical condition. That time this will divide, this will proliferate. So HSC, they are going to differentiate into LPC. LPC stands for lymphoid progenitor cell. Lymphoid progenitor cell will form the pro B cell. So what does pro stands for? Progenitor, okay, progenitor B cell. And during this step, what is the conversion, what is the new modification which is taking place is first heavy chain is formed, okay. So as we all know, in a B cell, okay, B cell is much alive, much similar. It is going to produce antibodies, okay. So this is the heavy chain, this is the light chain. So we have two heavy chains and two light chains. What happens is, in this step there will also be a production of surrogate light chains. Now what is surrogate light chains? Heavy chains, they are formed first, okay, and the surrogate light chains, they are actually the junction between the heavy and the light chains. They are going to support the heavy chain, okay, until the light chain is formed, because there is no stabilizing bond, okay, to support the heavy chain. Now after maturation it will go rearrangement, and the surrogate light chains, they are going to be replaced by the light chains. So progenitor B cell, it will be undergoing differentiation, it will form pre B cell. What is pre here? Pre stands for precursor B cell. And here the first chain is formed over here, second chain will be formed over here, and like I said, something known as surrogate light chain will also be formed over here. From pre B cell to immature B cell, by this time it will be developing two new light chains, okay. These are surrogate, these will undergo rearrangements and it will form light chains. So by this time the immature B cell, it will have two heavy chains, two light chains. Now it will also have something, these are the surface markers which will be present on the immature B cell, that will be CD40 and MHC class 2 molecules, okay. Now the first BCR that will be present, that will be formed, that will be MIGM, okay. You should remember this is very important for competitive exams as well. Now it is immature B cell right now, okay. Now at this point it will undergo something known as selection process. There will be something known as positive selection, there will be something known as negative selection. In positive selection, what happens is, it will be this cell, okay, it will permit the survival of only those B cells, okay, whose BCRs are capable of recognizing the self MHC molecules, okay. So those B cells that recognizes the the self MHC molecules, that will only be selected. So this is a chance event. Now they'll be given a second chance, okay. If in the second chance also they are not able to identify their own self MHC molecules, then they are going to be destroyed. So this is known as positive selection. In negative selection, there will be something known as, it will check, okay, how strongly it is going to bind. If the signal strength is very strong, the B cells, those B cells will be destroyed, okay, and those will be selected whose signal strength is weak. And here there is no chance of second chance and third chance, whereas in positive selection you can get a second chance, but after that you need the cell will the B cell, it will undergo apoptosis. What is apoptosis? Apoptosis is programmed cell death, and the cell will kill. It will be like it will kill on its own. It will be killed on its own. Okay. Now, so this is a selection procedure. Okay, which will go on. After that, the second BCR will be formed. First BCR was MIGM. MIGD will form. Okay. So after this formation, we will be confirmed that okay, positive and negative selection has already been carried out, and this will be formed after the selection process only. This won't be performed before the selection process. So now we have our mature or the naive B cells. Now what are naive? What does naive mean over here? It means that these are still inactive. They have not encountered any pathogen as of now. Okay, and they are going to reside in the primary follicular bone marrow and other tissues. So thank you.